Hello everyone, I welcome all to the 20th lecture of this course. This 20th lecture is on nanotoxicology. So this is the last lecture of this course. Okay. So till now we have learned various biomedical applications of these nanomaterials in this course, and today we are going to see the other side of nanomaterials, that is toxicity of nanomaterials. So in this lecture we are going to learn what is nanotoxicology, what are the various exposure scenarios, and what are the various methodologies available to study the toxicity of nanoparticles, and we are also going to learn what is genotoxicity and how to study the hemocompatibility of your nanomaterials, okay, and also how to study the in vivo assessment of your nanomaterials toxicity. So let us see what is nanotoxicology. So this nanotoxicology is the study of toxicity of nanomaterials, okay, and nanomaterials, even when made of inert elements like gold, become highly active at nanometer dimension. As you know that gold is a inert material, which we use it for making ornaments or jewels, and when it goes to nanoscale it will show you a different kind of properties. Okay. So here this nanotoxicology studies are intended to determine whether and what extent these properties may pose a threat to the environment and to the human beings. Okay. So here this nanotechnology is a double-edged sword. The same novel properties making nanoparticles attractive makes them potentially toxic too. Okay. So the particular care must be taken in nanomedicines, since in this area where greater exposure would be present. So most of the field have its own advantages as well as disadvantages. Similarly, this nanotechnology also like a double-edged sword. So it has its own advantages as well as disadvantages. So we have to select the nanomaterials wisely for the various biomedical applications. So why we have to study the nanotoxicology? You can see here these are the various consumer products where they are using these nanoscale materials. For example, so this L'Oreal company is using this nanoparticle based cosmetics, and Motorola is using the CNT based nano emissive displays, okay, and also we have nano care fabrics shirts and carbon nanofiber rackets, okay, and also in India also we are having several washing machine, okay, which is coated with the silver nanoparticle, and also in the nowadays in the TV advertisement we are seeing that some of the soap they are using silver nanoparticles, okay. So what happens, these silver nanoparticles and everything goes into the water bodies, okay. So when it goes into the water bodies, how it is going to affect the living organism in the water bodies, and again it will contaminate the groundwater, and how it is going to cause toxicity. So those things we don't know, okay. So in this lecture we try to learn what are the nanotoxicity methodologies available to study the toxicity of nanomaterials. So this you might have seen it in cricket, okay. So in the Australian players mostly they used to apply this kind of sunscreen, okay. So this is a bulk form of zinc which is white and opaque, okay, and this side he applied the same sunscreen, but it is in the nano zinc. So the nano zinc is transparent, and this bulk zinc, that is a sunscreen, it is a white and opaque, okay. So when the material goes to nanoscale, the properties of the material get changed, okay, and the nano gold can be red and blue, and carbon nanotubes can conduct electricity. So new applications for familiar materials, okay, and again but also new risk. So the inert material which we are using day to day, okay, when it goes to nanoscale it is going to have some different kind of properties, and different kind of properties it may have advantage, at the same time it may have new risk also. So these are the various companies involved in this nano. For example, Nestle, Pepsi, and some of the other companies are involved in this, okay. So most of the companies they product the product in the name of trade secret or in the name of patent protected laws, okay. So we are not aware about what are the nanomaterials in the food products. So these are the other products, nano based products, okay. For example, sunscreen, cosmetics, and even the baby diaper and baby bottle, feeding bottle. So everything they started using these nanomaterials and nano coatings. So but we don't know what will be the long term effects, okay. So this is in early 2008, approximately 104 foods and food additives contain nanoparticles, okay, which is on sale on internationally, and some of the analysts suggest that almost 500 nano foods are available in the worldwide, okay. So almost 500 nano foods are already in the market, so we are not aware about what are those, and there is no labeling. For example, if you are using the GMO food, so there should be GMO label. Similarly, if you are using a nano food or nano coatings, there should be some label to indicate these are coated with nanoparticles, or this food is wrapped with nano package. Okay, so there is no such labelings. And again, this nano can play a major role in agriculture also. So it can make the agriculture uniform, and it can make further automated and industrialized. Okay, and so due to which what will happen is the farmers may lose the farming knowledge, but at the same time, it can have the designer substance which can deliver the nutrients efficiently into the body. Okay, and these are some of the nano foods on sale, include cooking oils, teas, and also we can have the antibacterial kitchenware. Okay, and also food processing and food packaging material. So this nano based food packing materials will interact with the food, and it will prevent the microbial growth, but this nano material is also interacting with your food substance. So when it interacts with the food, how much amount of nanoparticle is releasing into a food material, and when you take those food, what will happen to you? So those things are not understood thoroughly. So recently a US based company, Nanotexicology, okay, so they made a slim shake chocolate. Okay, so this is a diet milkshake that uses silica nanoparticles coated in cocoa clusters to increase the taste with low cocoa and sugar content. Okay, but this health risk of nanosilica remain poorly understood, and early studies suggest need for caution. So this slim shake is made up of silica nanoparticles. So what is silica nanoparticles? Silica nanoparticles are basically sand particles. Okay, so in your childhood, if you eat a sand, your mom will beat you, but the same sand particles you are drinking in the form of silica slim shake. So but we don't know what will be the long term effect of this silica based chocolate drinks. Okay, and these are some of the other future nano food and agriculture, like we can have the edible nano wrappers and coatings. Okay, and also we can use the nano biotechnology to manipulation of seeds. So we can use these technologies to manipulate the seeds to increase the growth. Okay, and also we can increase the productivity, but again, what will be the drawback and long term effects still unknown. And again, this nanotechnology could enable junk food to be fat free, sugar free, and also it can reduce the carbohydrate, and it can increase the vitamin and protein, and it can be fiber enhanced. Okay, so then it can be marketed as healthy alternative. So this junk food can be enriched with protein and fiber, and it can be sold in the name of like a healthy alternative, but the problem is our relationship with the real food will be eroded. And again, this nano could displace the workers and erode the farming knowledge, because everything will be automated. So automated nano surveillance and management systems. Okay, so that could reduce the need for farm workers, and this nano could commodify farming knowledge, and the nano will play a major role, and it will take up the technologies like a proprietary technologies. Okay, so only some of the multinational companies, they can control the food and agriculture. So let us see some of the examples of how these nanoparticles pose some new toxic risk. So here the nanomaterials are readily inhaled and ingested, and at least some will cross the skin, and nanomaterials gain access to tissues and cells than the larger particles. Okay, and inhaled nanoparticles can also cross the blood brain barrier. Okay, so that is the important property of these nanoparticles. So most of the other drugs cannot enter the blood brain barrier, but these nanoparticles, it can cross the blood brain barrier. So let us see some more examples. So this nanosilver is toxic to rodent liver, brain and stem cells, and may harm beneficial bacteria also. And this nano zinc oxide is toxic to rat and human cells even at low doses, and nano silicon dioxide less than 70 nanometer can cause onset pathology similar to your neurodegenerative disorders, and this nano titanium dioxide can damage DNA in human cells, harm algae and water fleas, and especially with help of UV light exposure. So this titanium dioxide is everywhere in from your toothpaste and your talcum powder, and everywhere this titanium dioxide is there, and this nano titanium dioxide, when it go into the water bodies, and it is a very good photocatalyst in presence of sunlight, so it will act like a very good photocatalyst, so it can harm the algae and other living organisms in the water bodies. So here these nanoparticles can distort our immune system response, and here nanoparticles can also act like a Trojan horse, and the Trojan horse means like this nanoparticles will be in your body for more time, so it has more circulation time, and suddenly it may cause some new toxic effects. So that effect is called as Trojan horse effect, and this possible link between the consumption of processed foods and irritable bowel and Crohn's disease. So these nanomaterials can also induce these kind of diseases. Okay. So the UK Royal Society recommended in 2004, they made these are some of the rules for the nano safety. So that is a full safety assessment of all products that contain nano prior to market release, and all nano ingredients to be labeled, and environmental release of nanomaterials to be avoided as far as possible, and factories and research laboratories to treat nanomaterials as if they were hazardous. So let us see these properties of nanoscale materials. The same properties making nanomaterials so interesting can make them potentially harmful, as I told you earlier. So this enhanced reactivity and increased surface area to mass ratio and enhanced permeation, okay, and also previously unknown forms of common materials. So these are the properties which has a wide application in biomedical field, and also the same properties will also have some toxic effects. Okay. So let us see what are the various exposure scenarios. So all substances in the world are toxic to plants, animals and humans at some exposure levels. So there are two routes, short term routes and long term routes. So in the short term routes, it is mainly due to inhalation gas phase and skin contact in solution, and also oral ingestion, and in the long term routes mainly due to the soil adsorption and water dissolution and also biodegradation issues. So the risk evaluation for exposure to nanotechnology product is hindered by the law protected secrecy of product formulation, and also there is a lack of specific regulations on nanotechnology and non-mandatory reports on toxicity of products. Okay, and we are still using the old criteria, and methods became obsolete. And here these researchers from National Institute for Occupational Safety and Health so determines the concentration of nanoparticles in the air while unloading a reactor for producing the metal oxide nanoparticles, and these are the various international initiatives. Okay. So recently some agencies have taken some actions to establish regulations to nanotechnology. So International Standard Organization and US National Nanotechnology Initiative and British Standard Institute, okay, and also the Environmental Protection Agency. Okay. So all these agencies have published reports and guidelines related to the handling of nanomaterials and the research approach to nanotoxicology. So, but all of them are voluntary to follow. There is no mandatory rules and regulation. So let us see the methodologies for studying the toxicity of nanoparticles. So if you see, this is your carbon nanotubes, 100 grams of carbon nanotubes, but it look like a 100 kg. Okay. So there are several points we have to understand to study the toxicity of the nanomaterials. So what to measure, like how much duration and what is the route of exposure, and what is the dosage and formulation of test material, okay, and also what kind of biological species you are going to use to study the toxicity of nanomaterials, and whether do you have standard references and standard materials for comparisons. So these are the questions to be considered before we start the nanotoxicology studies, and also the main important thing is how to design a realistic test. Okay. So the meaningful results on the toxicity of nanomaterials are achieved when the conditions of possible exposure are reproduced accurately. So let us see how the nanoparticles would enter the body. The first one is accidentally, so it may be due to environmental contamination and workplace exposure. The next one is deliberately introduced, that is your nanomedicine applications. So different methods of exposure of the nanoparticle might produce different results. So let us see this with some examples. So the example one is installation of CNT in the rats. So here the rats that were instilled with high doses of single wall carbon nanotubes, it is died due to respiratory blockage rather than pulmonary intoxication. So here they used 4 kind of particles including single wall carbon nanotubes. By pressurized intrathecal instillation, the CNT was given to these rats, okay, and it was observed at 24 hours, 1 week, 1 month and 3 months, and here the results are like there is inflammation but no cytotoxicity, and this picture is showing that the respiratory airways are mechanically blocked by carbon nanotubes, due to which the rat was died. Okay. And the next example is inhalation of carbon nanotubes in rats. So exposing the rats to air contaminated with this carbon nanotubes led to the immune suppression. Okay. So in the previous example we have seen that when you inject these carbon nanotubes through intrathecal administration, what happens is that rat is died. Okay. So in this case, so we are passing this air contaminated with low concentration of carbon nanotubes, and exposure of 6 hours per day during 14 days, okay, and the tracking of proteins and immune response in the rats. So here the researchers observed that a signal like TGF beta is released when the carbon nanotube is inhaled. So this was tested by isolating the branchioalveolar lavage fluid protein from both exposed and controlled rats, and it was shown that the protein from exposed mice cause immune suppression. Okay. So here there is no death of rat, and here only the immune suppression. So from this example you can understand, so based on the method by you are giving the carbon nanotubes dosage to these animals, the results also varied. Okay. So when you instill these carbon nanotubes, the rats are died. So when the carbon nanotubes are inhaled by the rats, only the immune suppression happened. So what are the various test assessment? The methodology used led to different conclusion on the toxicity of carbon nanotubes. Okay. So several criteria must be used to select the meaningful results, like what test simulates a more realistic exposure, and also are these results applicable to all nanotubes, or exposure concentration realistic, and do animal subject respond similar to humans. Okay. So this reliable data to answer these questions can make difference between deeming these carbon nanotubes unsafe or safe. So what are the various methods available? Like we can use these in vitro methods. So in vitro methods means you can do it in the laboratory condition using the bacteria or cell lines to understand the toxicity, and in vivo means inside the living system, like living animal models like mouse or the zebrafish or some other animal models, and ex vivo is a combination of in vitro and ex vivo. So we can study the toxicity of nanomaterials using the bacteria and cell culture system in vitro. Okay. And we can also study the toxicity of nanomaterials using the animal models like a mouse or rat or zebrafish. Okay. So that is your in vivo. So that means in the living system. So this ex vivo is the combination of in vitro and in vivo. Okay. So we can take out the particular organ and we can grow in the lab, and we can study the toxicity of nanomaterials. So here the advantages are like it is cost effective, time effective, and no ethical issues when you use the in vitro conditions. And several studies conclude that response to nanoparticles is too diverse. For instance, dogs and pigs have a higher immune response to nanoparticles. Okay. And again this nanoparticles toxicity will depends on the formulation. So depends on what kind of nanoparticle you are using, what was the concentration, and also what is the pH, and what are the coatings of the nanomaterials, and also what are the exposure mode, exposure time, and which is the targeted organs. Okay. And what kind of animal model you are using, where are you going to use the rat or human cell lines or fish model, which model you are going to use, and what are the methods you are going to incorporate to understand toxicity, for example in vitro or in vivo or ex vivo methods. So depends on these 4 parameters. Okay, formulation, test, and subjection methods. So your nanoparticle toxicity will also vary. And we can also improve the in vitro techniques by substituting the 2D with 3-dimensional scaffold. Okay. So here these pores are the white circles. Okay. So these are created in a hydrogel matrix, and this is the liver cells are like a gray color circles. Okay. So the liver can be useful to study the toxicity of nanomaterials and in a 3-dimensional scaffold. So here the advantages are like it will represent the complex cell and cell matrix interaction, and also so with the help of tissue engineering we can develop this artificial liver, and we can easily understand the toxicity of nanomaterials. So in this the researchers have studied the toxicity of quantum dots using these 3-dimensional as well as 2-dimensional scaffold. So and they concluded that the 3-dimensional scaffold it's exactly mimicking the in vivo condition. Okay. So you can see here these red colors are dead cells. So as I explained in a previous lecture, so these red cells are dead cells. In the 2D with the same concentration you are seeing more amount of dead cells, and in the 3D culture you are seeing only few cells are dead cells. Okay. But the concentration nanoparticle is same. Okay. So when you use this 3-dimensional scaffold, and that will exactly mimic like your in vivo condition. And other alternatives like we can also do these modeling and simulation. The toxicity of nanoparticles depends on its physical and chemical interaction with the gases, liquids, and other nanoparticles surrounding them. So this can be studied using these molecular simulations. So the specific and the quantitative knowledge obtained from theory and simulation can help building the predictive models and algorithm for assessing the likelihood of toxicity in various natural environments. So if we know the toxicity of nanomaterials, so we can do the modeling and simulation, and we can accelerate this nanotoxicity field. Okay. So let us see what is genotoxicity. So genotoxicity refers to the ability of a test agent to induce DNA damage. Okay. So the cytotoxicity means if it causes toxic to the cells, okay, and genotoxicity is if it damage your genetic material. Okay. So here the genotoxicity assays like which can detect, quantify, and characterize the DNA damage induced by the substance under investigation, and these methods are indicators for likely carcinogenic agents, and the genotoxin means a substance that damage the DNA is called as genotoxin. So here this nanomaterial release free metal ions, so that will induce the oxidative stress, and this oxidative stress will damage your DNA, and it will induce the apoptosis, or it will induce the inflammation. Okay. So we have to understand whether this nanomaterial is inducing any damage to DNA or not. So you can see here when you expose the cells to these nanomaterials, what happens is like there may be chances for chromosomal aberration. So if it is a normal chromosome, there is no toxic effect, and if you are identifying that there is a chromosomal aberration, that means it is due to this, it can leads to abnormal cell growth, and it may be, the compound may be tumorigenic, and we can also understand the genotoxicity by studying the DNA double strand breakage, okay, and by using this various nucleus stain and cell division and proliferation. So these are the various toxic methods available. Okay. So we can use this Salmonella typhimurium to understand whether this nanomaterial is inducing any mutation in your genetic material. Okay. So that is assay is called as Ames assay. Okay. And also we can use it cell lines, mammalian cell lines, to understand whether it is inducing DNA laddering, and we can also understand the apoptosis and everything. So using this MTT assay and other apoptotic genes. Okay. So this I already explained in the previous lecture. So let us see what is the Ames test. Okay. So here we can use a bacteria called Salmonella, okay, and which requires histidine for its growth, and to this we can add the possible mutagen. In this case we are using the nanoparticles. So this bacteria will be grown in a media with a minimal histidine, okay, and in presence of this possible mutagen, if it causes a mutation, it will cause this histidine negative to histidine positive. Okay. So this bacteria will grow more on this plate. So if the bacteria growing more on this plate, that means the nanoparticle may induce some mutation in the genetic material. So you can see here when compared to the control plate where only few colonies were there. Okay. So let us see what is hemocompatibility assay. So because the hemocompatibility is very important factor to decide the application of implantable biomaterials such as artificial blood vessels and orthopedic implants, and with the development of blood contacting materials or implantable devices, it is necessary to improve the hemocompatibility by surface modification or redesign. So whenever you make nanomaterials or nanoparticle based coatings, so we have to understand whether it is compatible with the human system, and also we have to understand whether it is compatible with the blood. Okay. So by using this hemocompatibility assay, we can understand the compatibility of your material with the blood. So the hemocompatibility test of nanomaterials contain the following assays. So it is a hemolytic assay or anticoagulant assay, and platelet adhesion and activation assay, blood coagulation time assay, and blood protein adsorption assay. So how to collect the blood? We can take the blood from human. Okay, you can take a 2 ml of blood. Okay, or you can collect the blood from the goat also. But in case of human, when you collect your own blood also, so you have to take the ethical committee permission, human ethical committee permission. So it is easy to take the blood from the goat. So you can collect the blood from the nearby slaughterhouse, and you can use it for hemocompatibility studies. So here when you collect the blood, you have to mix the blood immediately with the tripotassium EDTA, that is your anticoagulant. Okay. So this blood can be, the goat blood can be collected from the slaughterhouse, and in presence of anticoagulant. Okay. So then you can centrifuge it at 13,000 rpm for 10 minutes at 4 degrees Celsius. So when you centrifuge, you will get this kind of layers, and you can use this isolated platelet rich plasma, that is PRP, for your further studies. And here this is your polymer film, and you want to understand whether this polymer film is hemocompatible or not. Okay. So you can cut into circular disc, using this even a simple punch machine, okay, sterile punch machine. So it can cut a uniform size, and this can be sterilized by keeping it under the UV light for 30 minutes, and this can be transferred to the 96 well plate, okay, and washed with PBS. So then you can add the isolated platelets on the polymer circular disc. So you can add 200 microliter of PRP in each well, and you can incubate in the incubator for 30 minutes at 37 degrees Celsius, okay, and you can observe the adhered and activated platelets on the polymer disc surface using the microscope, and you can wash this 3 times with the PBS to remove the non-adhered platelets, okay. So when you view under the microscope, you will get this kind of results. If you are getting this kind of result, that means less adherence of platelets, okay, and there is no change in the shape. That means there is no release of coagulation factor, and it is a good material for implant. So under the microscope, if you are seeing this kind of shapes, that means more adherence and the change in the shape. So it is mainly due to the activated platelets. It is releasing more coagulation factor, and it is not good material for implant, okay. So next one is in vivo assessment of nanomaterial toxicity. So why we have to use zebrafish as a human model? Because it is a small vertebrate tropical water fish, okay. So it has a clear and transparent embryos with a short maturation time, and functionally homolog with almost 70% of human disease genes, and forward and reverse genetic model system is possible here, and we can also do the chemical biology and chemical genetic studies using zebrafish. So let us see some of the examples how we can study the toxicity of cellular nanoparticles using these zebrafish embryos, okay. So here they, in this paper they have used 2 different kind of nanoparticles, okay. One is starch coated cellular nanoparticles, one is BSA coated cellular nanoparticles, and we can study the toxicity of the nanomaterials in terms of mortality rate of zebrafish or hatching, as well as the heart rate and abnormal phenotypes, okay. So this is a zebrafish heart beating, okay. So we can measure the heart rate also compared to the control, what is the heart rate in the treated zebrafish embryos. So here you can see here, so they have used that zebrafish embryo and treated with a different concentration of cellular nanoparticle. So this is our normal embryo, and this is a malformed embryo, and these are the dead embryo in presence of cellular nanoparticle. So based on that, you can count the number of embryos which got the malformed embryo as well as the dead embryo, and based on that you can calculate the toxicity of nanomaterials. And in this case they have observed that the cellular nanoparticle is in the most of the mitochondria as well as the nucleus. That means it is possible that the nanoparticles enter the cells through many routes. Among them, endocytosis through the embryo wall is more likely, okay, and it is entering the mitochondria as well as the nucleus, and it may induce some damage to your DNA also. So by elemental analysis of embryo, they have observed that cellular nanoparticle is more in the nucleus. So they believe that it may create a cascade of toxic events which may lead to damage the DNA, okay, and which will leads to various toxic effect. Zebrafish embryos with a different concentration of cellular nanoparticles. So you can see here this heart rate is going down, and again this mortality rate is increasing, okay. So this nanoparticle deposition in the central nervous system could have adverse effects in the control of cardiac rhythm, respiration and body movements. So you can see here this heart rate is going down with respect to concentration, and again this exposure to cellular nanoparticles resulted in the accumulation of blood causing edema and necrosis. So that is why this mortality rate is increasing. And we can also use that mouse or rat model for studying the toxicity of nanoparticles. So Organization for Economic Cooperation and Development guidelines recommend oral toxic test and dermal toxic test to understand the toxicity of nanomaterials. So for the oral toxic test, colloidal nanomaterials at the limited dose of 5000 milligram per kilogram body weight could be given to the mouse, okay, and it can be observed for toxic symptoms for the first 3 hours and after 24 hours. So these animals could be maintained and observed daily over 14 days for the skin and also behavioral symptoms and toxic sign, and then we can do the hematological and the biochemical testing. So these animals will be sacrificed after 14 days, and histopathological testing could be made for understanding how much is nanoparticle deposited in the kidney, spleen and liver. So next one is the dermal toxic test, okay. So we can apply these nanomaterials on the surface of the animal models. Okay. So, the group 1 is treated with the distilled water, and the group 2 receives 50 ppm of colloidal nanoparticle solution, and group 3 we can use a 100 ppm of colloidal nanoparticle solution. So, we can increase the concentration depends on the study. So, when you treat with these different kind of nanoparticles, then you can study the hematological and biochemical analysis and also the histopathological investigations to understand the toxicity of nanomaterials. Okay. As a summary of this lecture, in this lecture we have learned what is nanotoxicology and what are the various exposure scenarios, and also we have learned what are the various methodologies available to study the toxicity of nanoparticles, and we have also learned what is genotoxicity and also how to study the hemocompatibility of your nanomaterials and in vivo assessment for studying the nanomaterials toxicity. So, I will end my lecture here. I thank you all for listening to this lecture and all the best for your exams.